that they also have a lot of well-regulated promoters. Hopefully you can differentiate between the different promoters of different expression systems, and you can also correlate that with whether it's tightly regulated, what type of basal expression, and so on. We also talked a little bit about complementation selection. So this is, for example, if you have a string and a vector which has leucine 2 wild type versus leucine 2 mutant, and how you can actually use that to select for your recombinant protein of interest. We also talked about proteins in vascular virus insect cell expression. So this was pretty brief. This was when you have your insect genome together with your transfer vector. Usually your transfer vector contains your gene of interest, and when there is recombination, it's able to update your gene of interest. So pretty simple system, also known as a binary eukaryotic expression system. And we also talked about mammalian cell systems. So this one has a eukaryotic promoter, and then hopefully you can remember how that differs from the bacterial promoter, and polyadenylation, and protein affinity tag. So we also talked a little bit about how you can use the tag to be able to get your protein of interest, whether based on size or charge, among others. And then lastly, we ended with workflow of cloning expression and purification of recombinant protein. So that includes the exact workflow starting from cloning all the way to purification. Any questions before we move on to transgenic animals? Everyone good? Okay, cool. Lots of thumbs up. Alright, so for transgenic animals, we will have 3 different classes. The first one today will learn about the experimental basis of transgenic animals. So what that basically is why would we actually even need transgenic animals in the first place. So let's move on to question 1. You can join through Slido, or since we are actually face-to-face, if you just want to raise your hands and shout out the answer, you are also welcome to do that. I'll give you guys a minute to type out your answers. Are there any transgenic animals in Asia? Otherwise, why have we talked about all this recombinant protein and all these different things? Have they actually been applied in Malaysia? And if your answer is yes to this question, do you know what those transgenic animals are? If you think your answer is no, why do you think the answer is no? I'll just wait for a couple of answers and then we'll proceed to the next slide. Welcome to those who just joined. So just a quick question to gauge, I think so. Someone said no, someone said yes. Lots of yeses, lots of no's. Animal tissue culture is not encouraged in Malaysia. Interesting answer. Yes, is it a pig? I just guess. Okay. I like that answer. So someone took an educated guess. Transgenic fish. So I'm guessing this person might have looked at the lecture slides. Yes, someone said mice. Transgenic fish. No, maybe a yes. Okay, yes. Yes, sold as food. Modified lactose. No, because of ethical issues. No, yes. Yes, but restricted to special labs. I'll probably take the last 2 answers and then we'll move on. Yes. We'll just wait for the last one. Hopefully, yes. No, Malaysia's too far behind. That's a very sad comment. Okay, so on that note, I'll quickly give you guys the answer to this question. There actually are transgenic animals in Malaysia. There are some animals which have been imported. For the ones who answered mice and whatnot, mice, there definitely is a lot of mice that has been brought in for work in labs. But the people who answered things like there's not ethical issues and whatnot, that is also true. So in some instances, transgenic animals that were made elsewhere, it's easier to bring them in than to actually make transgenic animals in Malaysia. But that said, there actually are transgenic lobsters, transgenic prawns, and I think transgenic fish that actually have been made in Malaysia. You will probably hear from the scientists who made those animals. At the end of the semesters, I'm trying to bring them in to give you guys a talk, so you guys will actually get to hear from them directly what their journey and how they actually managed to come up with this transgenic animal. So hopefully you guys are looking forward to that. Thank you so much for the answer, guys. Alright, so when we talk about transgenic animals, obviously there's lots of different things to consider. Even just based on your answers earlier, you see things like people talking about it in the context of using it in lab, people talking about it in the context of maybe as produce, because someone said, using it in being sold in markets and whatnot. But something that was sort of really interesting was in 2003 when they actually made transgenic fish that glow in the dark. So, just out of curiosity, if someone sold you transgenic fish that glows in the dark, who would actually consider buying it as a pet? For what to eat, is it? So you said, I would rather eat it. Who would buy it as a pet? Okay, a couple of people. Right, so what was interesting was that it was definitely approved by the US to be actually sold as a pet. But when you actually approve usage as a pet, which is not directly related to medical purposes or something where there's actually an actual essential need for it, it actually caused a lot of panic and worry among other people. Because if you do this, how do you know what else will be genetically modified? So that sort of did set the, platform or the stage for how people actually view transgenic animals or recombinant animals. But when we do talk about transgenic animals, we do sort of have to go back and look at some of the history of how this actually came about, and that actually traces back to the 1960s. So over the next couple of slides, we will talk about some of the early experiments that were actually crucial to identifying what are some of the key components as to how you can actually make recombinant organisms. So on the screen here, you see the Griffith experiment. It's called the Griffith experiment because it was actually done by a scientist called Frederick Griffith. And if you look at the image on the left, so they started out with two different strains of bacteria. So the first strain of bacteria is a non-virulent strain called the R strain. So if it's non-virulent, shouldn't cause any damage, it shouldn't cause any hurt to the animal. So when they actually injected it into the mouse was healthy. Next, they used a different strain, an S strain, which is a virulent strain. So if it's a virulent strain, and you inject it into the mouse would die. So far, everything's pretty clear, right? Okay, cool. What they did next was, they then heat-killed strain S. So if you heat kill a strain, if you heat kill a bacterial strain, is the bacteria still alive? Yeah, see some heads shaking no, the ones in front shake no. The ones at the back, if you heat kill a bacterial strain, is the bacteria still alive? The ones at the back. Yes or no? Who says yes? Okay, so one of the answers is depends on the bacteria. This is how you know there are lots of microbe students in the group. So one of the answers is it depends on whether it's a thermophile, so it is able to sustain itself in a high heat. But in general, when you talk about a heat-strained bacteria, it does mean that if you inject it into the mouse should still be healthy. Making an entrance. Alright, so then the last experiment that they did was they actually took the non-virulent strain, strain R, they took the heat-killed virulent strain S, and they injected both into the mouse. So if you look at them separately again, the non-virulent strain R, the mouse should be healthy. The heat-killed virulent strain S, the mouse should also be healthy, but when you actually put them together, the mouse dies. But what was also more interesting was when they actually took a sample of what's actually in the mouse, they actually found out that there was actually live strain S bacteria found in the mouse. Everyone can follow the experiment so far? Okay, cool. So this was very interesting because it actually did suggest that there is a possibility for living cells to be able to take up surrounding genetic material. It's a lot like if you have bacteria A, it's on the floor, if it encounters genetic material from bacteria B, it's able to take up material from bacteria B. So this is a concept that's known today as transformation. So if you guys remember what we did in lab last semester, you guys actually learned the concept of transformation in the context of lab. So this was very, very important because again, other than the concept of transformation, it actually also told people that possibly DNA, RNA, or protein is an important hereditary material that can be transferred to another organism to give you a different genotype or phenotype. So in order to determine whether it's DNA, RNA, or protein, every McLeod and McCarthy did this experiment. So what they did was they started with the heat-killed S cells again, the one that was found in the very last mouse. They divided it into 3 different tubes. So the first tube, they added proteinase. So when they added proteinase, that means there should be no protein in the first tube. In the second tube, they added ribonuclease, which means that there should be no RNA in the tube. And the last one, they added deoxyribonuclease, which means that there should be no DNA in the tube. And then they added R cells back to the different beakers and the tubes, just to check to see whether any S cells appear. So S cells actually did appear in the tubes that didn't contain protein or didn't contain RNA. But for the tube that didn't contain DNA, no S cells appeared. So all this is to say that DNA is what is crucial in order for transformation to happen. So if you don't have DNA, you'll see what happens at the very last beaker, which is no transformation occurs. Everyone okay? Okay, cool. Alright, so let's talk a little bit more about what are some of the other key developments in recombinant DNA technology. So 2 other important experiments, or 2 other important concepts is one is the concept of cut and paste. So cut and paste, very simply put, is if you have sort of 2 different vectors and you want to take a component from one vector and transfer it to the other, you cut it from the first vector and then you have to be able to paste it into the second vector. So I'll go over that in detail in the next slides. And also, you transfer foreign DNA into new cells. So we saw that a bit earlier, transformation, which happens in a natural setting, but how do you actually manipulate it so you can actually do it in the lab? So the image on the right, so again, since most of you guys were in lab with me last year, you'll remember you started with a plasmid, it underwent a transformation process, and then you grew it up in agar, and all your agar had colonies. Right, so this goes into the whole cut and paste concept a little bit deeper. So in this case, again, if you cut a piece from vector 1 and want to put it in vector 2, you have to be able to glue it together. That glue is known as ligase. There's a lot more other things that is important, but basically something that's really crucial is that it needs to be compatible. So if you look at the image again, if you look at what's in red, you see that the sequence is CTTAA, and the one in blue is AATTC. So it needs to be compatible, which means that the sequences actually need to complement each other, and that's actually how you get to ligate the products together. So if you have 2 ends which are not compatible, so say one sequence is AAA, the other sequence is GGG, because they don't complement each other, even if you add ligase into it, there's no way you can combine both those products together. And then of course, in order to actually cut a certain region from a vector, we use what is known today as restriction enzymes. So restriction enzymes will give you that overhang that you saw on the slide before, that AATTC, I think. So depending on what restriction enzymes you cut your vector, your plasmid with, you will get different ends, which will then enable you to combine those pieces together. This slide itself, it just sort of shows you how restriction enzyme was first discovered. It was first discovered in bacteria because it was actually used to prevent the growth of bacteriophage, which would attack the bacteria. So then they actually realized that how it worked was that when there were these restriction enzymes, it would actually cleave the phage DNA at certain sequences. So for example, if it cuts at AATTC, it would always cut at AATTC. So from there, that's how today we have restriction sites and restriction enzymes. And there are probably more than a few hundred different restriction enzymes out there. Questions so far? Right. So once you have cut and placed different components together, you want to introduce it into a new host cell. So something that people did in the early days in the 70s was that, again, if you look at the image on the left, on my left, you'll see that you have an EcoR1 treated plasmid enzyme. So EcoR1 is a restriction enzyme. So they can cut the plasmid at different places, and then if you just sort of throw them all together, because they all have the same sticky ends, if you add ligase into it, that means you can get any combination together. Does anyone need me to repeat that? No? Okay, cool. So when you do that, that means that, again, you can get a combination of C and D, C and E, C and F, and so on, just because you have all of them with the same restriction enzyme and you're just randomly adding ligase into your mix. From there, in this case, they are using calcium chloride to treat the bacteria, which is often used as a transformation step, and then you will get bacterial colonies floating on the plate. From this as well, what they realized was that some bacteria actually had antibiotic resistance factors. So this is again something that you learned from last semester as well. And when you do have something like an antibiotic resistance factor, when you transfer it into, or introduce it into a different cell, that cell then will also have antibiotic resistance. So the person who discovered all of that in the slide before, his name was Cohen. He was the first one to actually construct a novel DNA plasmid using two separate plasmids. And again, he used restriction enzyme. Today, this is done very, very commonly. So if you want to take a promoter from plasmid 1, you want to take antibiotic resistant gene from plasmid 2, you want to take a different tag from plasmid 3, you can do all of that. So you can actually come up with any different plasmid that you want and subsequently transform it into a bacterial cell and then use it as a recombinant protein. So when you use different restriction enzymes, all those sequences are unique, and again, they have to complement each other before you can actually ligate them together. Right. So this was actually also another interesting experiment. So in a lot of the other experiments that we saw, they often only combine DNA sequences from similar organisms. So if you're using bacteria A, they would use bacteria A and then add it to a different sequence of bacteria A. But this experiment was important because in the 70s, Paul Berg, another scientist, took phage DNA, he took an E. coli DNA, and then he took an SV40, SV40 is a virus sequence, and he actually combined them together. So he actually showed that you are able to combine the DNA of 3 different very different organisms together. So it was quite a big thing. So that the image on the left is what he made back then. But when he realized that he could do this, in the 70s itself, he actually suggested a moratorium on all recombinant work. So a moratorium is when we just want everyone to stop doing any sort of work. I see some very confused faces. So the reason why he wanted a moratorium was because he realized that if you are able to make something like that, which is a combination of phage, virus, and bacterial DNA, anyone can come up with anything. And his worry was, what if someone comes up with something extremely dangerous? So if there are no rules in place, how do you actually ensure the safety of scientists or people who might have to deal with the repercussions of all these recombinant plasmids that people come up with? Alright, so quick question. Again, I think this is the last question for today's class is, when we think of the moratorium, so it was put in place because he wanted people to stop doing something dangerous. So how might transgenic animals be used wrongly? What sort of transgenic animal might people come up with, or what can people possibly come up with things that might potentially be dangerous? Or even if you don't want to answer it in terms of transgenic animals, just anything at all, can you think of what people might do where it's just wrong or not okay according to you? So even if your answer is, I don't like blue strawberries, why would anyone want to come up with blue strawberries? That's also okay. Maybe to you, blue strawberries are the most awful thing to look at. You can also answer that. So I'll give you guys a couple of minutes. Lots of answers already. Okay. So lots of bioweapons, biological warfare, transgenic insects that destroy crops. Actually, that's quite an interesting answer. Animals that are aggressive, but really enough to be used for war. Harmful for eaten by Eugene. Yes. When we eat transgenic animals, more meat, attack humans. So it looks like a lot of you turn into zombies. Producing animals with high concerns and strength to be used as weapons. Okay, see a couple of people still typing, so I'll give it like another second. Reduce nutritional value of food to contaminate human genome, poisonous meat. Yeah, so yeah, still 3 people typing. I'll wait for the last one and then I'll quickly talk about it. Disrupt ecological balance, dangerous food to be consumed by humans. Threat to Aboriginal. So cool. Thanks so much for your answers, everyone else. Okay, cool. So if people don't know what Lulu and Nana is, so Lulu and Nana are actually genetically modified humans. They were the first two genetically modified twins who were modified to make sure that they were resistant to HIV. So that was actually probably the pinnacle of what can possibly go wrong in
Chinese scientists
ethical aspects when it comes to people. So cool. Very, very interesting answers. So even as all of us as scientists, we
edit
can
the
easily
genomes of
imagine
human embryo.
what we're planning on doing.
可是你要 start from 它还是 embryo 的时候，如果它不是 embryo， 那个人了。
Disadvantages if you combine DNA together, or if you decide to do something that is
Who is responsible for something?
Thank you so much for your answers. It's very cool answers. Didn't know. I don't know if this person means that they remember me transgenic as well. We will talk more about that later, and you will see what you guys say about that. Alright, so that was in the 70s, but in the mid 70s itself, actually a lot of, in 1975, So 3 or 4 years after the moratorium, a lot of scientists actually got together and they actually put together a lot of laws and regulations pertaining to recombinant DNA. I think it's in 1975. And those rules and laws are actually still used by everyone globally even today. So in the 80s itself, there were 2 other scientists when they introduced rapid DNA into murine germline cells, and that was the first recombinant animal ever made in the 80s. Alright, so this is the second half of the class. Awesome, I'm right on time. Any questions so far? No, everything crystal clear? Cool. So in the second half of the class, we will mostly talk about the application of transgenic animals. I'm sure most of you already know this, so there'll probably be more revision, and we won't get into the technicalities of it until our next class. Right, so recombinant DNA obviously is used to create transgenic animals. So some of the very early transgenic animals were goats and cows with milk modification to make allergen-free milk. Today, there's a lot of other animals where the milk has been modified. So not just to make it allergen-free, but to make sure it is more nutritional, that there's antibacterial properties and whatnot. So we'll see examples of that later. There's also been lots of disease-resistant chickens that's been made. Again, we'll see the details of that later. In your class later in the semester, we'll talk about genome editing as well. Ruben and Anna was genome edited. And we also learned that with that, you can add any new genes and modify animals in any way that you want. Alright, so one of the most important reasons why people wanted to work with transgenic was so that they could actually make transgenic livestock. Again, transgenic means when you are actually able to introduce DNA sequence into the genome of a cell. Hopefully you guys can see the image on the left. So in order to make a transgenic animal, usually you start by designing your gene construct. So your gene construct is very similar to what you've seen in the plasmid sequences. You source the transgene, so you actually get a company to make the plasmid. Today, it's very easy for you to make the plasmid yourself. You transfect it into certain cells. You select for the cells. So again, if you have certain selection markers, you can make sure you're selecting cells that have your recombinant DNA. You make a transgenic embryo. So in our next class, we'll learn how to make transgenic embryos and the pros and cons of different methods. And then you finally confirm that your animal is transgenic. So again, in our next class, we'll learn about the pros and cons of all the methods listed up there, whether it's retrovirus-mediated gene transfer or germ cell transplantation. When we talk about a transgenic animal, it specifically refers to an animal where they are able to pass on the transgene to its offspring. So something for all of you to think about is that a good or a bad thing? Would you want a transgene to be passed on to offspring, or would you not want an animal to be able to pass on the transgene to an offspring? Something to think about. Alright, so in general, with transgenic animals, they usually use it to study the genetic control of physiological systems, to build genetic disease models, to improve production traits, and to produce new animal products. All this would significantly improve human health. So some of you did point out correctly, if you do use it the wrong way, it would be detrimental to human health, enhance nutrition, protect the environment, increase animal welfare, and decrease disease. So again, these are the general applications of transgenic animals in agriculture. So if you look at the image, maybe 30 years ago, people mostly used nuclear injection or nuclear transfer, so they would have their recombinant DNA, recombinant protein, introduce it that way, and then they would have their transgenic animals. So in our next class, we'll learn how the different methods that I talked about, which one actually has a high success. So if your success rate is something like 1 in 10,000, which means that for every 10,000 embryo that you try injecting, only one is successful. Is that very good or bad thing? see lots of people shaking their head now. So we'll discuss all that in the next class. So again, practical applications of transgenic is mostly for reproduction, growth rate, disease resistance, and modifications of traits. So earlier on, I think a lot of people commented that they probably wouldn't want to eat any transgenic animals. At least that was what I saw in the exercise. I think that's what you guys were saying. But if we have things like a change in climate conditions, which means that we no longer have good food security. So I don't know if anyone remembers, a couple of months ago, I think KFC didn't have any chicken. I see a couple of little nodding. So if this were to actually continue, and KFC doesn't have chicken for the next 2 years, would people be okay with transgenic chicken? Who would be okay? Oh, okay. Who would definitely not be okay?
It's all right.
So for KFC, everyone would be okay. so it is really sort of dependent on what's going on in the world right now. And it's not just chicken. Right now we also see a decrease of eggs. There hasn't been eggs anywhere in the country for a long time. There are also things like, when we talk about climate change, so we are seeing different weather patterns right now. So more rain or less rain, which also affects things like what crops can grow. And like Cameron Highlands, I think right now, I think they were saying it's 2 degrees hotter, which means that everything that used to come from Cameron Highlands probably won't be available anymore in a couple of years. So probably no more strawberries. I see some sad faces. So in instances like that, would you guys be okay if you have genetically modified strawberries? Alright, so just things to think about for you guys, especially as scientists. How do you actually determine what is necessary and what is important to actually be made into a transgenic crop or livestock? Alright, so moving on a little bit. Thanks guys for participating in my random questionnaire. So of course it is for enhanced nutrition. This is seen quite a lot, especially in fish. Because you can modify fish and other animals to have better quantity of food, better quality of food, and specific nutritional composition. So fish is used quite a bit because it is able to enhance omega-3 fatty acid, which is consumed by humans. So again, if you have something that's lacking in your natural dietary resources and you are able to make transgenic animals, which can improve human health, then some people think, why not? This is another thing that is quite commonly done, again, mostly in fish, but also in other livestock as well, is enhanced growth rate. So if you look at the 2 fish at the top, so those are the non-transgenic fish. If you look at the 2 fish at the bottom, that's the transgenic fish. So that actually is with transgenic, you actually are able to cause a 30 to 40% increase in growth rate. Can anyone think why this might be important? Why do you care if a catfish is tenfold heavier than what it used to be? Or why would you care if a fish is 40% larger than what it used to be? Can anyone think of a reason? I can hear some answers, but you guys will have to shout. At the back, I think. It's more meat, okay? Or you can just eat 2 fish, right?
There's gonna be scarcity, that's why I'm going for transgenic.
Okay, so that's a good answer, thank you. So yeah, so if you don't have sufficient fish, then yeah, you probably want to be able to have 1 fish which is a lot larger, or if it's more expensive, you probably just want 1 fish which is a lot larger, which does tackle again food security problem. Any other reasons that anyone can think of? Here's some answers, but I can't really tell who's answering. Probably have to shout it out again. This group over here is discussing very seriously. Do you guys have Okay, yes. No need, still want to think about it again. Okay, cool. So, but yeah, that's a good answer, right? What if you do have scarcity? And it's not just in terms of fish. If you have things like, say, floods happen more frequently now, say instead of just at the end of the year when it's a monsoon season, say now floods happen every 6 months. that's quite bad because that will also affect production or the availability of different fish. So if you do have an enhanced growth rate, it probably is quite beneficial. This is another common one, enhanced milk for better quantity and nutrient content as well. So you will see with a lot of transgenic livestock and crops, it's mostly for better quantity and higher nutrition. So you do get things like increasing certain proteins in the milk, which will help impact growth, development, health, and survivability of cattle, sheep, and goat. So if you look at the image up there, you will see that there's a huge increase in meat production because of the availability of transgenic animals. Even things like improving your milk quality also affects the effect on the offspring, which subsequently will lead to better meat and so on. So this is just a very brief summary. If anyone is interested, you can just Google and look up papers on how they did this. But there's tons of transgenic animals that have been done so far. So they have animals which express all of that in the milk. So whether it's lysozyme or lysostapin, bovine, beta, alpha-lactalbumin, lactoferrin, and under those promoters. Right, so enhanced disease resistance is also a big thing. So in the example up here, I talk about mastitis and inflammation of mammary gland, which is caused by pathogens, which causes a decrease in milk production. But obviously, you can also think about how, in general, if there are particular pathogens which are affecting certain animals, and this is commonly seen, especially if all animals are of a certain breed, or if all animals came from a certain farm. So if you are able to make them disease resistant, then you are able to ensure that the quality of your meat is good and that not all the animals are with chicken especially, so it's not just the meat, but also the eggs, for example. There's also transgenic dairy cows that secrete lysostatins, and this is also in the slide before this, which kills staph aureus. And they've also made livestock which are prion-free. Prion is not so much a big problem now, but a couple of years ago when there was mad cow disease, this was a huge problem, and they actually had to cull a lot of livestock. So today they're actually able to make prion-free livestock. Okay, any questions so far? There's still a bunch of other examples. No? Okay, all good. Right, so improved animal welfare is also a big thing, especially in Western countries. So, for example, if you talk about chicken, in certain countries they actually chop off the beak of the chicken and they actually chop off the wings of the chicken, because assuming you have to grow 1 million chicken in this classroom space, if you chop off the beak and you chop off the wings, then you can put more chicken in the classroom space. This is actually a real thing. Yeah, it's quite sad. Likewise, for cows and calves, if you have horns, like big horns, they also take up a lot of space, so they actually chop it off as well. So that is actually what is done. So there's actually fault cows, skirt cows, and dehorn cows. So for a long time, dehorn cows were what was commonly done. Again, it was mostly because people were trying to grow a lot of particular livestock or animals in a small space. So that is obviously very difficult for the animals. So what they've done is they've made a lot of transgenic animals where they just grow horn-free, like the polled cow over there. So if they're horn-free, then you are not chopping off the horns of the cows, and that way it is better for the animal welfare. Right, there's also improved reproduction. This is when you have manipulation of certain genes, such as estrogen receptor genes. I won't talk about this too much. You can ensure that when you make a certain animal, you are selecting for certain animals which are able to produce more offspring. And that's again something that's quite useful, because if say in 1 year you're able to make 10 cows, versus in 1 year you're able to make 100 cows, anything that gives you 100 cows would be better. also usage of improved hair and fiber in certain transgenic animals. In this case, they can manipulate the quality, learning strength,
I did the
and print of wool and hair fiber from sheep and goat. So a lot of people like to use the hair from sheep and goat for various purposes. You've probably seen certain carpets and certain clothes made from that. So if you are able to make transgenic goats with spider silk, then you can imagine that the wool or the fur or the hair will probably have a different consistency, a different texture. So to some people that's quite priced. You actually can use it for medical devices, airbags and clothing as well. In Malaysia, there actually are people who are trying to modify or genetically manipulate spider silk. The other purpose of transgenic animals is for bioreactor. Bioreactors usually used to produce human biological products. For example, blood-derived human serum albumin, HSA, is prescribed for liver failure and traumatic shock. Sometimes when there is a shortage of blood supply, so HSAs typically come in blood. So if there is a shortage of blood supply, especially like what we saw throughout the pandemic, there's actually a massive shortage of blood everywhere because no one could actually go into the hospital to donate blood. So if you have transgenic pigs, which actually have more HSA in blood, then you actually are able to tackle this problem. Obviously, with all the examples that I show up here, it's things that have been done, but something you can think about as a scientist in Malaysia is if this is something that may not be relevant to Malaysia, can you replicate this in a different animal? Right, almost to the end. So there's also reduced environmental impact. So there are also certain livestock which can impact the environment based on the carbon footprint and sort of what is the byproduct of it. So pigs in general don't consume enough dietary phosphorus, and because of that, you actually have higher levels of phosphorus levels in their waste products, and it causes pollution problems. So this is typically seen around farms which breed pigs. So pollution is bad, and they make transgenic pigs which express phytase, which actually is able to allow for better digestion of phosphorus. And when you have better digestion of phosphorus, that means you actually have a reduction of your waste products and subsequently a reduction of pollution. Alright, second last slide. This is probably one of the most useful advantages in transgenic animals, has been used for a long time. But transgenic animals are made, so what we pointed out, mice for example, because they make suitable animal models in biomedicine. So there are a lot of things that maybe we might want to study in humans. For example, maybe we want to study something about the human brain, but we obviously can't go around and cut open someone's head to study their brain. So, but what we can do is maybe we can make a transgenic animal which mimics something similar that we want to study in the human brain. So, for example, if you want to see how cancer affects the brain, you can come up with a transgenic mouse that has cancer and see how that affects the brain. So this has been used a lot. One of the best uses was when they actually used a sheep knockout model to mimic deafness, which was actually pretty cool. And also for xenotransplantation for organ to prevent organ rejection. All right, last slide. So as usual, I'll just end the lecture with questions to think about. So can all recombinant systems be utilized to produce transgenic cells? So just a heads up, usually all these questions would probably be somewhere in a test or an exam or something like that. So something to pay attention to. So can we use all recombinant systems to produce transgenic animals, or can we not? So for example, we think of mammalian recombinant systems work, but not virus recombinant systems. What are some of the key early experiments that influence the production of transgenic animals? We learned about the and experiments. Do you think there's something else that actually has made it possible to make it? Do you think that recombinant paths are necessary? Carl, you voted that yes. love to have glow-in-the-dark fish. Probably some of you might also like to have glow-in-the-dark cats. I don't know. But are they necessary? Should we have? Hear some people saying yes. They would be so cute. So should we have transgenic animals that are not beneficial medically, but is physically attractive? So maybe they don't help with nutritional value or they don't necessarily help with increase of need, but you just want something to look at because you're stressed out from your FYP and you want to go home and look at cute animals. What are some other functional transgenic animals that we mentioned? So earlier we discussed what would we not want, but what is something that we don't have in Asia or in the world that's sort of lacking that might be addressed if we have those transgenic animals? What is the cost of producing transgenic animals like transgenic animals? What would be the cost of producing transgenic animals? Goodness. Okay, so something that if any of you do continue doing science, whether it's a master's or a PhD, or continue doing research in the future, you'll probably think of a lot of solutions to address these problems. If your solution costs, say, $1 million, would that be worth it? Something to think about. Or, do you always want something that is cheap? Or do you think your cutoff is like, as long as it's 100 ringgit and below, that's okay? And then finally, what is the main limitation factor of transgenic animal production in Malaysia? So again, for those who sort of came later, we will have an invited speaker who has worked on transgenic animals and crop and livestock in Malaysia. At the end of the semester, I think I'm still trying to find the best dates, so you probably get to ask them as well, sort of what were the challenges when you first started the work. Right, so I think that's it for any questions from anyone?